Dear students, I hope you all will be fine. I'm here, Muhammad Jamal Khan, lecturer in botany, University of Education, Lahore. Today, we will discuss a topic from one of the core subjects of botany, that is plant systematic anatomy and development. The course code of this subject is BOTN 1112. So here we have the ultrastructure of cell wall. Basically, ultrastructure is the architecture of cells, and it is a biomaterial that is visible at higher magnification and that found on a standard optical light of microscope. So it's a fine structure, especially within a cell, that can only be seen with high magnification microscope. So basically, these ultrastructure is the result of arrangement of special molecule or the building blocks of cell wall, that is cellulose. So the special arrangement of cellulose forms specific structures, and these structures to referred as ultrastructure of cell wall. As you know, by the arrangement of cellulose, it produces a middle lamella, primary and secondary wall of cell wall. In addition to this, these structure, primary wall, secondary wall, these are basically formed by the combination of different cellulose molecule in the form of macrofibrils, microfibrils, and Micelles or elementary microfibrils. So here we have these building blocks of secondary and primary wall that we categorize as given below in our next slide. So here you observe, this is a simple wood. This is a woody tree, and when we observe the structure of this wood, these are the wood cells as you observe. So after deep observation of these wood cell through a high magnification power, we observe that these cells are further comprised of specific substances or the specific arrangement of cellulose that may form microfibrils or macrofibrils or microfibrils, and in the end it produces a specific structure, that is elementary microfibrils or Micelles. So these structures are elementary microfibrils or Micelles. So here I have a dramatical representation of these ultrastructure referred as microfibrils, macrofibrils, and Micelles or elementary microfibrils. So initially we start here we have in A we have a cell wall that is composed of primary cell wall. Then we have a secondary cell wall. Secondary cell wall may be further comprises of 3 layers, S1, S2, S3. We already studied it in our previous lecture. Then we have a middle lamella, that is a cemented layer that join the two cells with each other. So from this we start. This is the section of secondary or primary wall. So from this, here we have the building blocks of this secondary and cell wall, that is microfibrils. Basically these microfibrils composed of cellulose. So cellulose arranged in a specific manner to form a macrofibril. Then these microfibril is further revealed that there are microfibrils that combine to form macrofibrils. So here we have microfibrils, and after this, after further revealing, we observe that here we have the cellulose molecules that arrange into a specific pattern, and these cellulose molecules we call it as micelles, or we also call it as elementary microfibrils. So elementary microfibrils are the cellulose molecules. Basically, we start these building blocks of primary and secondary cell wall from descending order. First, we discuss the micelles or elementary microfibrils that combine to form microfibrils, and then these microfibrils combine to form macrofibrils, and the microfibrils united together to form a primary or secondary wall or cell wall. So for your elaboration, again I have a pictorial animation for you. Here we have the cell that is so here we obtain the cell wall. So this is the cell wall, and this cell wall is composed of cellulose, and the arrangement of cellulose further categorize it into fibers. Basically, cellulose arranged into fibers. So these fibers may be a macrofibrils or maybe a microfibrils, and these microfibrils is further classified into Micelles. Or these Micelles is also called as elementary microfibrils, and these elementary microfibrils basically composed of cellulose, lignin, and hemicellulose. So basically these Micelles or elementary microfibrils is also referred as cellulose nanocrystals, and these crystals actually combine to form microfibrils, and the microfibrils combine to form macrofibrils, and macrofibrils combine to form a fiber. So this is the descending order. Here we have the descending order, and we discuss later in our this lecture. Here we have some features of elementary microfibrils or Micelles. So basically these microfibrils or Micelles in which the cellulose molecules are combined into many very fine bundles, and these bundles are called Micelles, or you can also say that elementary microfibrils. So the size of these microfibrils are near about 100 angstrom in diameter. So basically the Micelles or the elementary microfibrils are crystalline aggregate of cellulose molecules, and these Micelles basically are individual units. So each micelle is composed of 14 cellulose molecules, and most of them are arranged in crystalline order. It means it is composed of crystals, and only a small part also contains small also arranged in randomly to form a paracrystalline structure. So a short arranged range of crystal is referred to as a paracrystalline order. In addition to this, only some micelles also contain small amount of chitin, as in case of algae, and the interfibral matrix contains a specific compound, that is pectin compounds, and hemicellulose that is play a very vital role for the arrangement and for the attaining the specific pattern of the arrangement of cellulose molecules. In addition to this, the addition of these substances, as I already told you, like cellulose, like hemicellulose and pectin compounds into cellulose fiber, this is called as incrustation. So by doing this, one can increase the tensile strength of the cell wall, and this is also the reason due to which the lignification occur in higher plants. Next, we have microfibrils. So Micelles or elementary microfibrils combine to form macrofibrils. The size of the microfibrils is near about 250 angstrom, and the microfibrils basically are the basic unit of cell wall, and the orientation of these microfibrils determines the nature of cell wall. These fibrils have a circular or longitudinal or oblique arrangement, and these arrangements determine the tensile strength, plasticity and elasticity of the cell wall. So basically the arrangement of microfibrils, because these are the basic unit of cell wall, play a very vital role or perform a function of their plasticity, elasticity and the tensile strength of the cell wall. So here we have a figure of cellulose microfibrils. Here you observe clearly in this slide, these are the microfibrils. These all are microfibrils, and these microfibrils arrange itself into a specific pattern, and this specific arrangement is just due to the presence of pectins. And then we have the semicellulose. These structures are semicellulose, and these are structural proteins that is indicated by pink color, and these thread-like structures or black thread-like structures are called pectins. So by using these structural proteins, pectin and hemicellulose, these microfibrils arrange itself into a specific pattern to form the next structure, that is macrofibrils. So here we have another view of microfibrils. So these are the pectins indicated by this strands. These are the pectins. These are the pectins. In these pectins, a blue shaded indicate a specific compound that are hemicellulose, and in addition to this, we have these red rods, small rods. These red rods, I change the color. So these red rods, that is referred to as cellulose microfibrils, and these light pink or purple color, these are the glycoproteins or proteins. So these are the structures that involve in the combination of microfibrils. So in the last we have macrofibrils, and the macrofibrils actually are the result of microfibrils, because the microfibrils combine together to form a large size bundles called macrofibrils, and the size or the diameter of these macrofibrils is 0.4 micrometer. Here we have a diagram. These small structures, these are basically microfibrils, and the microfibrils combine together, number of microfibrils combine together to form this macrofibril, and this microfibril combine to form a fiber of cellulose or a cell wall. So basically these microfibrils or elementary microfibrils or micelles combine to form a cellulose chain. So here it clearly indicates these are, as you see, these are the cellulose beta glucose molecules. These are the beta molecules in cellulose chain, and several hundred chains are present in it, and then these combine to form a microfibril. Here this one microfibril contains number of micelles and elementary microfibrils, and these microfibrils, number of microfibrils combine to form a macrofibril. So actually these are the arrangement of cellulose molecule into a cellulose chain. Now the next ultrastructure we discuss now, that is Plasmodesmata. So basically Plasmodesmata. So here I have a pictorial demonstration for you. Here we have two cells. This is the cell number 1 and this is the cell number 2. In between these two cells, here we have a this specific ultrastructure, that is Plasmodesmata, that is indicated here also. So this one is cell 2 and this one is cell 1, the cell membrane of cell 1 structure. So here we have these cytoplasmic extensions or strands, that is referred as Plasmodesmata. So when we observe through a microscope at 53x part, here we observe clearly these Plasmodesmata that observed through this magnification. So basically these are the cytoplasmic extensions, or these are the strands that help the cell for transfer of material or substances through each other. So here we have another view. So these are the cells, cell 1, cell 2, and in between those cells we have cytoplasmic strands or extension that indicated by this. These are the Plasmodesmata, and here we have the view of these Plasmodesmata by TEM microscopy that you observed here in this pictorial view. So basically the Plasmodesmata are cytoplasmic strands that extend through the pores in adjacent cell wall. Here we have another demonstration related to Plasmodesmata, and this demonstration is better represent the Plasmodesmata. So here we have the cells. These are the cells. You observe these are all our cells. Here we have group of 4 cells, 1 and then 2 and then 3 and then 4. So here in cell number 3 and 4 we observe the Plasmodesmata. So here these are the adjacent cell wall of the plant cell, this one and this one. In between these two plant cells, here we have desmotubule that is passing through the Plasmodesmata. So this one is a Plasmodesmata, and this is cell number 1 and this is cell number 2 for your elaboration, for your understanding. So when we observe this Plasmodesmata through microscope, here we indicated in this form. These are the hollow spaces or structures. These in between these we have a specific structure. These one, this one, and this one, and this one. These are Plasmodesmata, and the size of this opening or a cytoplasmic strand is 0.6 micrometer. So here we have some of the functions of Plasmodesmata. It helps in the transportation of material. It also regulates the traffic of molecules from the adjacent cells, and it also play a role in the development of organelles, and most of the viruses also move through this cytoplasmic extension of strands, that is Plasmodesmata. The next ultrastructure of cell wall is pits. So simply we defined these are the cavities or thin areas within the thick secondary wall are called pits. Basically pits are left during the process of secondary wall formation, and such cavities or perforation are also found in the primary wall. So these pits actually are the result of non-deposition of lignin or cellulose. Due to this, these cavities or openings or perforations are produced. Such cavities are referred as pits. So when these pits are found in primary wall, we called it as primary pit field. One of the role of primary pit field is that the plasmodesmata are formed by these primary pit fields. So the primary pit fields are turned into pits during the secondary wall formation. But pits are also arised at place where there are no primary pit fields. So it's not means that the pits are depend upon primary pit field. So the pits of the adjacent cell are complementary pits. Due to this complementary nature, we call it as complementary pits. In this case, pits of cell wall of two adjacent cells are in front of each other. The two opposite pits are separated by a thin layer of secondary wall that is called as pit membrane. A pair of opposite pits in the wall of adjacent cell is referred as pit pair. Sometimes a cavity is formed by the break in the secondary wall that is called as pit cavity. The opening of the pit on the inner side of the cell wall is referred as pit aperture. The membrane built of the primary cell wall and middle lamella that separates the two pit cavities of the pit pair is referred as pit membrane, as I already told you. For your illustration, I have a pictorial image for you. Here you see these arrow indicates these are pits, or it indicates the presence of pits. So how these pits are formed? Here we have the primary pit fields, and these fields formed during the secondary wall formation, and these primary pit field combine to form plasmodesmata. So basically these primary pit field are present in cell wall. Here we have the primary wall, middle lamella, and due to the action of these primary pit field, it creates plasmodesmata. So these two pits, this one and this one, these are pit pairs because they are adjacent to each other. In addition to this, we have this small line that is here. This one is pit membrane, and the opening of this aperture of this pit, we called it as pit aperture. So here we have, this one is a pit aperture, this one is a pit membrane, and these are the pit pairs. And this hole is called as pit chamber, or it is also referred as pit cavity. So here we have different types of pits. We have simple pits, border pits, half border pit pair, and then we have blind pits and compound pits. We discuss later in this lecture one by one. As you observed in this image, a simple pit. Basically, the diameter of this simple pit cavity remains uniform, and the pit cavity opens in the lumen of the cell. Therefore, the pit of two sides of the common wall form a simple pit. So here we have another image of simple pit. Here we have the surface, indicates a surface view, and this one indicates the sectional view. So these are the simple pits. These are simple pits that are uniform, in the cavity, and also to adjacent cell. So here I have another type of pit, that is bordered pit. So in this case, a secondary wall arches over the cavity of it, and thus the pit opening become narrow than the pit diameter. So this opening is small as compared to this diameter. So the border pits of the opposite cells form a bordered pit pair. So this indicate here, and this indicate this actually produce a border. So due to this, it is called as bordered pits. So the border of pit open in the cell lumen by pit aperture, as I already told you about pit aperture, and the pit aperture is circular and much narrower than the diameter of the pit chamber. So in certain cases, the pit aperture are lenticular or linear or oval, depend upon the type of plant, or depend upon the type of secondary wall formation. So the pit membrane of the border pit develops an oval thickening in the middle, and this oval thickening is referred as torus. Here, so this one is a torus. So pit membrane is flexible, and the liquid in cell put pressure on torus membrane. So pit membrane pushes the torus, that the torus closes the border pit, and it shows that torus control the passage of pit through border pit. So the border pits are more complex than simple pits, and they are found in vessels, tracheids, and fibers of xylem. The next type of pit is half bordered pits, as you observe in this figure. So in this case, border pits has a complementary simple pit, and such a pit pair is called as half border pit pair. So here we have a border pit, and instead this one is the border pit, and in front of this border pit we have just a simple pit. So such type of pits we called as half bordered pit. So the other type of pit is blind pits. So here I draw a blind pit. So basically this pit do not have any complementary pit. So due to this we call it as blind pit. Here I indicate you by this red line. So here we have this one. Here we have a this one is a pit. This is the pit aperture as indicated here. This one is the pit cavity, and this one is the pit membrane, this, and this one is a torus. So in front of this pit there is nothing, or it haven't exhibit a complementary pit to the adjacent cell. So such type of pits we call it as blind pits. So here we have the last type of pit, that is compound pits. In this pit sometime it occurred that there is one pit on one side. Here only a one pit, this one is only single pit, and but there are two or more complementary pits on opposite sides. So here we have one, and this one is two pits here. So on one side we have a single pit, and on the other side we have two or more than two complementary pits on opposite sides. So such type of pits are referred as compound pits. So on the base of the arrangement of pits, we further classify the pits into three forms. Here we have secillariform, opposite, and alternate. In secillariform, in this case the just pits are arranged in transverse rows, while in opposite, in this case, pits are on one line and in pairs, and in alternate, in this case, pits of two linear rows alternate with each other simply. So this is all about for this lecture. Thank you.